everyone. So I have a terrible, awful, dreadful final on Monday, and I just can't study for it. Like I not that I can't. I can't. It's quite a pain because it's really important and I really want to, but for some reason I get distracted and it's hard. So because I like making videos, I've decided to film myself while I'm going through the slides and the lectures because I think that's something I've never done before. Pretty sure I've never done that before, and it would probably work best for me right now. I have so many things going on. I'm trying to put this first, but it's hard. So let's hope this works. Now I'm gonna be looking at my slides and talking about them. I don't expect this to be like a lecture. I mean, it's not gonna have pictures or anything. But I'm hoping that the information that I'm telling about is gonna be useful for somebody somewhere. So if you do want to see the slides that I was looking at, just message me and I'll send them to you. I just can't put them on here because I don't have the rights to, so. But I can talk about it, so that's what I'm doing. Alright, so here we go. I'm going to start backwards. I don't know why I'm doing this, but I'm going to start with the last lecture that's going to be on the test, and that is Archaea. Oh, by the way, this is a microbiology class I forgot to mention. It's a microbiology lecture. It started off pretty basic, but it got really complicated really fast. So hopefully this will help me do a lot of progress. It's not like I'm not gonna fail or anything, but I'm an overachiever and I really want to do it right. So here I go. We are talking about Archaea. Archaea were long mistaken for bacteria because they do have many similarities, which we'll talk about, but mainly shape and form. I mean, under the microscope, they look like bacteria, so people thought, oh, they're bacteria. Well, they're not. Not all of them. There are things that make them similar to bacteria, like I said. For instance, they're small. That's, good reason. Then they also have no membrane surrounding their nuclear material. So basically they don't have a nucleus, they have a nucleoid like bacteria do. One more similarity would be the circular chromosome. And all of this was discovered by sequencing of the 16S rRNA. And this put Archaea on a different evolutionary path than bacteria, and that will raise even more questions. One thing about this class that I don't like is there's so many unanswered questions. Like we ask our professor very interesting questions and he can't tell us the answer because he doesn't know. Things are just so cryptic. It's annoying. Alright, let's talk about some unusual shapes. Archaea do have unusual shapes. I'm looking at these pictures and I don't know what I'm looking at. I see some flower-looking things. I see diamond-shaped bacteria. Well, they're not bacteria. Well, diamond-shaped organisms. And then there's ones that I swear looks like a chicken. But I'm probably wrong. And then there's the one that looks like a Y. Again, if you want to see all of this that I'm talking about, just send me a message. And then we get to the classification of Archaea. There are three groups. We have hyperthermophiles, those that live at over 100 degrees Celsius. You can find those in Yellowstone or in deep-sea hydrothermal vents, as we'll talk about later. Then we have extreme halophiles, which love salt. And those you can find in California or in obviously Great Salt Lake, Utah. And our last group are methanogens. I don't know how you pronounce that. Methanogens, I guess. And those you can find in Poland or in Siberia and places like that. Some things that make Archaea different, though, because they are a different evolutionary group altogether, is their cell wall. Bacteria have peptidoglycan in their cell wall, which is altogether special itself. But these Archaea have a pseudo peptidoglycan. Ooh, what does that mean? Well, it's similar in function, it does the same thing, but structurally it's different in that it has no N-acetylmuramic acid. Instead, it uses this unpronounceable thing called N-acetylallosaminuronic acid. Oh, that's why I love science. And it doesn't use L-amino acids, but it uses it uses L-amino acids rather than D-amino acids in cross-linking. Yay, fancy. One more thing about the structure of Archaea is the wall's made entirely out of protein, which I don't think I've seen before in this class, but here. So these are called S-layer protein shells. They are a crystalline shell of glycoproteins, basically proteins that are decorated with sugars. And they provide structural integrity like any wall would do. One thing that distinguishes Archaea furthermore from bacteria is the ether-linked lipids. That's what happens in Archaea, but in bacteria they are ester-linked lipids. It's chemical difference. I'm probably going to forget this, but I said it, so maybe if I watch this sometime I'll remember. At the same level of the lipids, Archaea have extensive branching, crazy branching, whereas bacteria have maybe one single branch at the end. They're not crazy about the whole fixing solubility. And this makes us think that elongation during lipid synthesis in Archaea is quite different. Like I said, in the bacteria, branching is rare. It does increase molecular space in the membrane, and it increases membrane fluidity. But bacteria don't need to alter with that a lot. They're pretty versatile, so no need for it. But in Archaea, there's extensive branching, and this obviously decreases molecular space and shape, and decreases membrane fluidity quite a lot. Oh, let's see, what else? Some Archaea further limit molecular motion with a membrane monolayer. Now I'm looking at this picture trying to figure out what it means. Membrane monolayer. Alright. I believe... Oh. Okay. You know how bacteria has a B layer? Well, they have a monolayer. It accounts for extreme membrane stability of hyperthermophiles. I'm looking at the picture. Oh, okay, never mind. I was going to say I don't get it. But I do. In the membrane bilayer, you have, the phospholipids that are coming together in the opposite directions to, hydrophilic versus hydrophobic. God, I just can't talk today. So there are two layers, but in Archaea they have the monolayer, which is just one single layer. I know how stupid that sounds, but it makes sense maybe if you see the picture. But I'm sorry, I tried.